Welcome back to the deep dive. Today, we're really getting into the weeds, you could say.
Huh. Quite literally, maybe.
Yeah, we're looking at the engine room of modern plant science, gene discovery and validation. It's, incredibly complex stuff.
It really is. Think of it like finding that one specific, crucial needle in a gigantic genetic haystack.
And that needle, once you find it and you're sure it's the right one, that's key for everything that comes next, right?
Absolutely. Whether you're designing a new transgenic cassette or planning, like super precise genome editing, it all starts there.
So our mission today is to give you a clear map, a structured way to understand how researchers actually do this, how they bridge that gap between pure genomic data and, well, actual plants in the field.
Exactly. And that gap is the core challenge, isn't it?
It seems to be. We've got decades of genomics generating just mountains of data.
Unbelievable amounts. Sequence data, variations.
But then you have traditional breeding, which is all about practical results, yields, field performance, and sometimes it feels like they struggle to connect.
That's a great way to put it. See, genomics is often chasing causation, finding the statistical link, the why behind a trait. Why is this plant taller? Why is it drought resistant?
The association.
But the breeder, they want the application.
Mm-hmm.
How do I use this improved genotype? And sometimes that statistical link genomic finds, well, might just be a marker that's nearby, not the actual gene doing the work.
Oh, okay. So linkage, but not necessarily the direct cause.
Precisely.
Yeah.
Which is exactly why validation is so critical.
But okay, before we get into the how, let's ground this. It all comes back to the central dogma, right?
Yeah, but-
The absolute foundation.
Always. Can't escape it. Information flows, DNA, the gene,
Gets transcribed into mRNA.
which then gets translated into the protein, the workhorse molecule.
And it's those proteins doing the actual job. A tiny, tiny change back at the DNA level.
Like a single letter swap, a SNP.
Yeah, that could change one amino acid in the protein.
And that single change might tweak an enzyme's efficiency, maybe alter a whole metabolic pathway, or it could change a transcription factor.
Flipping a gene switch from on to off or dialing it up or down.
Exactly.
Yeah.
And that's what causes the difference we see, the phenotype.
So how did we first start finding these genes systematically?
Well, the first big wave back in the early 90s was expressed sequence tags, ESTs.
Right, EST sequencing, a quick snapshot of what's being expressed.
Yeah, a way to quickly survey the active genes. But now sequencing costs have just, well, plummeted.
So we moved way beyond short ESTs.
Oh yeah, the whole genome sequencing. Much faster, much cheaper for, major crops like rice or Arabidopsis.
And after you get all that sequence data, you have to assemble it somehow.
Right. Computationally. You cluster all those fragments, align them, get rid of redundancy, and you end up with what we call unigenes.
A single non-redundant reference set for the genes.
That's the bedrock.
Okay, foundation laid. Now, the first big hurdle. Pinpointing a candidate gene. What exactly defines a candidate?
Good question. Simply put, it's any gene that, based on some initial evidence, looks like it might be related to the trait you care about, your phenotype, or maybe the disease resistance.
And researchers tend to split these candidates into two broad types.
Generally, yeah. The first are positional candidates. Here it's all about location, location, location.
Like real estate?
Kind of. You identify them based purely on where they sit on a chromosome. Maybe you found a quantitative trait locus, a QPL, in that region, or you're doing map-based cloning.
Okay, so position on the map points you there. What's the other type?
Functional candidates. Here, you suspect a gene because its known biological function seems relevant to your trait.
So if you're studying, say, starch content.
You'd look for genes already known or predicted to be involved in starch biosynthesis, often using tools like transcriptomics to see which genes are active.
Got it. Okay, let's unpack the main strategies for actually finding these positional or functional candidates. Two main paths, right?
Two main philosophies almost. First is forward genetics, phenotype first.
Meaning you start with an interesting observation in the plant.
Exactly. You see a plant that's doing something interesting or useful, maybe it's super salt tolerant, then you work backwards to find the bit of DNA responsible for it.
And the classic method here is map-based cloning.
The real classic. You find your mutant phenotype, you genetically map where the mutation roughly is on the chromosome.
And then the hard work starts.
Oh yeah, fine mapping. They call it chromosome walking or landing. You narrow down the region bit by bit until you physically isolate the gene sequence. It's laborious, Definitely tedious.
Yeah.
But, you started with a phenotype that matters.
Okay, that's the old school. What's the modern high-throughput version?
That would be Genome-Wide Association Studies, or GWS.
GWAS. I hear that term all the time.
It's powerful. You take a whole collection of different plant lines, often quite diverse, maybe unrelated.
Like a diversity panel.
Exactly. You sequence them, or at least genotype them, for millions of DNA markers, usually SNPs.
Single nucleotide polymorphisms.
Right. And then you use statistics to look for associations between those SNPs and the trait variation you're measuring across the panel.
And that gives you those famous Manhattan plots.
That's the one, the skyline graph.
Why Manhattan?
Because if you plot the genome position along the bottom and the statistical significance, like the p-value, going up, the spots with really strong associations shoot up like skyscrapers against the baseline.
Ah, okay. So the tall buildings point you to the interesting neighborhoods on the chromosome.
Pretty much. High peaks mean strong statistical links in that genomic region.
Now, there's a clever twist on this mapping idea, right? Bulked segregant analysis.
BSA. Yes, it's a really smart modification of the older QTL analysis. Saves a ton of sequencing.
How does it work?
Instead of genotyping maybe hundreds or even thousands of individual plants from across.
Which costs a lot.
Right. You just take the plants showing the most extreme phenotypes, say the 20 tallest plants and the 20 shortest plants in your population.
Okay, the extremes.
You pull their DNA, mix the DNA from all the tall ones into one tube and all the short ones into another tube.
So just two samplings?
Just do bulk samples. Then you sequence only those two bulks. They call it BSA-seq or sometimes QTL-seq.
And compare the sequences between the tall bulk and the short bulk.
Exactly. You look for SNPs or other variations that are like fixed differently between the two bulks. Those variations must be in the region controlling the height difference. It's a super fast way to narrow down the location.
That sounds incredibly efficient. But what about the variations in the middle? If you only sequence the extremes, are you potentially missing genes with smaller effects or maybe useful alleles that aren't just tall or short?
That's the trade-off, absolutely. BSA-seq is fantastic for finding major genes quickly, big effect stuff.
Right.
If your trait is really complex, controlled by lots of genes, each with a tiny effect, a truly quantitative trait, then yeah, you might still need a full GWAS or a detailed QTL map.
Okay.
But for nailing down major trait genes fast, maybe for initial screening, BSA is a huge time and money saver.
Okay. That covers forward genetics starting with the phenotype. Let's flip it. Reverse genetics.
Right. Genotype first. Here you start with a gene, maybe one you suspect from the literature or a previous study.
And you mess with it.
You mess with it. You alter its sequence or maybe its regulation, and then you look to see what happens to the phenotype.
How do you mess with it?
Well, the classic way is mutagenesis. You can induce random mutations using chemicals or radiation.
The scattergun approach.
Yeah, expose a whole population and then screen for interesting new phenotypes.
Mm-hmm.
Or nowadays, it can be very directed.
Like with CRISPR.
Exactly. Using something like CRISPR-Cas9, you can target a very specific gene, make a precise cut or change, and then see if the plant changes in the way you predicted.
What other reverse genetics tools are common?
RNA interference, or RNAi, is a big one. It's like a dimmer switch for genes.
How so?
You introduce specific small RNA molecules into the cell. These trigger the cell's own machinery to find and degrade the messenger RNA from your target gene.
So you silence the gene, or at least knock down its expression.
Right. And if silencing gene X causes the phenotype you're interested in to appear or disappear, that's pretty strong evidence that gene X is involved. Functional validation.
Okay. And one more. Tilling.
Tilling. Targeted induced local lesions in genomes. It's kind of a hybrid approach.
How does that work?
You start by creating a large population of mutants, usually using chemical mutagens like EMS, which typically creates point mutations, single base changes.
Okay, so random mutations again?
But then you use high throughput methods to specifically screen that population for mutations within a known gene you're interested in.
So you target your screening to a specific gene even if the mutation was random.
Exactly. Originally, they used enzymes that cut mismatched DNA, but now it's often coupled with sequencing. It's a great way to find a range of new, maybe subtly different alleles for a gene you already know, potentially creating novel phenotypes.
Okay, so we've got these sophisticated forward and reverse strategies finding candidates. But then comes the data avalanche. Millions of sequences, polymorphisms. How do you even begin to make sense of it all?
Yeah, welcome to the 21st century challenge. That's where bioinformatics becomes absolutely essential.
It's more than just storing data, right?
Much more. It's
Yeah.
a connective tissue. The umbrella that integrates information across all these different omic platforms we'll talk about, it's what creates that direct link
Yeah.
between the genetics and the actual plant phenotype you see.
But managing the data itself is a huge task. We have these massive public databases.
Right. The big three: GenBank in the US, EMBL in Europe, DDBJ in Japan. They've been coordinating since the 80s, trying to standardize how gene features are described, annotated.
Using a common feature table format.
Yes, trying to create some common language. But then
Then came the explosion of species-specific databases.
exactly. Databases for sweet potato, cassava, maize, rice, you name it, plus larger platforms like Phytosome or Gramine that try to cover many species.
Which sounds good, more focused information.
It is good in one way, but it also creates, well, data silos. Different formats, different priorities. It actually makes it harder sometimes to integrate data across species or platforms. It reduces the scope for big picture analysis.
So we have fundamental tools like BLAST.
The basic local alignment search tool.
Yeah.
Absolutely crucial for taking a new sequence you just found and quickly seeing what it looks like, comparing it against everything known. Essential for annotation.
But if the underlying databases are fragmented, even BLAST has its limits.
It's a constant challenge. The tools are powerful, but making the data play nicely together is tough.
Yeah.
However, there's been a really big conceptual shift recently in how we think about genomes. The pangenome.
Okay, pangenome. Heard this a lot too. What's the idea?
For decades, we relied on a single reference genome for a species, like the human genome or the rice genome, usually based on one specific lab strain.
Right, the standard.
The pangenome concept says, wait, that single reference doesn't capture all the genetic diversity within the entire species.
So it includes genes found in some individuals, but not others.
Exactly. And not just single gene presence absence, but also big structural variations. Large insertions, deletions, inversions, copy number variations, things that are often missed or poorly represented when you only looked at one reference.
Especially in complex, repetitive regions of the genome.
Precisely. The pangenome aims to capture the entire set of genes and genetic variations present across multiple individuals within a species. It gives you a much richer picture of diversity.
That must significantly change how we think about what's core to a species versus what's variable.
Absolutely. We now talk about the core genome, the set of genes found in pretty much every individual, highly conserved essential functions.
And the dispensable genome.
Also called the variable or accessory genome. These are the genes or sequences present in only some individuals or strains. And often this dispensable part is enriched for genes involved in things like stress response, disease resistance, adaptation to specific environments.
Really important stuff for breeding.
Hugely important. It highlights variation that was previously hidden.
This idea of looking across individuals and species also relates to comparative genomics, doesn't it? Using evolutionary relationships.
Yes, exactly. Comparative genomics leverages synteny.
Synteny being the conserved order of genes on chromosomes between related species.
Right. Evolutionarily, blocks of genes often stay together even as species diverge. So we can use COS markers, conserved orthologous set markers.
Genetic anchor points.
Kind of, yeah. They mark genes that are clearly descended from a common ancestor in related species. These COS markers allow you to map equivalent regions between, say, tomato and potato, or sorghum and maize.
So what's the practical benefit?
Well, imagine you find an important gene in a well-studied crop, like tomato. Using synteny and COS markers, you can make a really good prediction about where the equivalent gene, the orthology, is located in a less studied relative, like eggplant, even if eggplant isn't fully sequenced yet.
So it lets you transfer knowledge between species, a shortcut based on evolution.
Exactly, a very powerful intellectual shortcut.
Okay, so we've used forward or reverse genetics to find a candidate. We've used bioinformatics, maybe pangenomes and comparative genomics to locate, annotate, and organize it. What's the final critical step?
Proof, functional confirmation. You need to show that this gene actually does what you think it does at a molecular level. And that brings us to the omics toolkit.
Omics, genomics, transcriptomics, proteomics, metabolomics.
All the omics. We need high-throughput data from multiple levels of biology to really nail down function.
Starting with the message, transcriptomics.
Right. Looking at gene expression globally at the mRNA level, which genes are switched on or off and how strongly under different conditions.
They used to use microarrays or macroarrays for this, sticking bits of DNA on a chip.
Yeah, immobilizing known DNA sequences, cDNAs or oligonucleotides,
Oh,
and then probing them with labeled copies of the mRNA from your sample, say, treated versus control tissue. Differences in color or intensity showed you differential expression.
but now,
Now, the dominant technology is RNA-seq. You just sequence the mRNA population directly using next-gen sequencing.
more powerful, cheaper.
Much more powerful, better dynamic range, can find completely novel transcripts, and, yeah, often more cost-effective now. It gives you a really detailed snapshot of the transcript, all the RNA molecules.
Okay, so we know which genes are being expressed. What about the actual workers, the proteins?
Proteomics?
Proteomics looks at the protein level, identifying the proteins present, their abundance, their modifications, sometimes even their structures and interactions.
Databases like SwissProt have been around for ages cataloging protein sequences.
They have. SwissProt started way back in '86. But proteins are, well, trickier than DNA or RNA.
Why is that?
They fold into complex 3D shapes. They get modified after translation, phosphorylation, glycosylation, which massively affects their function. Quantifying them accurately at a large scale is still challenging.
But crucial, because proteins are what actually perform most cell functions.
Absolutely vital. Modeling the proteome and how proteins interact is a huge area.
Okay, DNA, RNA, protein. What's left? The end products. Exactly. Metabolomics. This focuses on the small molecules, the metabolites.
Sugars, amino acids, lipids, secondary metabolites like pigments or flavor compounds, starch.
All of those. Metabolomics measures the levels of these compounds, often using techniques like mass spectrometry or NMR,
Why is this considered so important?
because it's often the closest omic level to the actual phenotype you care about. If you want a sweeter tomato, you measure the sugars. If you want higher oil content, you measure the lipids. It's a direct readout of the plant's biochemical state.
And we have databases like KG that map out biochemical pathways.
Right, the Kyoto Encyclopedia of Genes and Genomes. KG and others give us the known roadmaps for how these metabolites are produced and interconnected. Metabolomics lets us see the traffic flow on those roads.
So bringing it all together, genomics, transcriptomics, proteomics, metabolomics, that's systems biology, right? Trying to understand the whole picture.
That's the goal, the holistic approach, trying to understand how all these different layers interact to produce the final outcome, the behavior of the system as a whole.
Which is more than just the sum of its parts.
Always. And the big challenge here is integration. How do you make sense of data from all these different sources?
How do you do that?
One key development is using standardized vocabularies like the gene ontology or GEO terms.
GCOs provide a common language.
Exactly. They provide consistent descriptions for gene functions, biological processes, cellular locations. So a gene annotated in a genomic database can be linked to expression data or protein interaction data using these common GEO terms. It helps bridge the gaps.
And this complex web of interactions across data types is sometimes called interactomics.
Yeah, interactomics is often used to describe the study of these interactions, whether it's protein-protein, protein-DNA, or integrating across multiple omic levels.
Wow. Okay, so stepping back, we have these strategies, forward and reverse genetics to find the genes. We have bioinformatics and pangenomes to organize the massive data. And we have the omics toolkit, transcriptomics, proteomics, metabolomics for functional proof. What does this whole complex pipeline enable for the future of breeding?
It enables things that honestly sounded like science fiction just a couple of decades ago. We're talking about really ambitious goals,
Look what?
like pathway-driven breeding. Instead of just selecting for yield, actively re-engineering entire metabolic pathways.
The classic example being C3 to C4 photosynthesis conversion.
That's the big one. Taking a C3 plant like rice, which is less efficient in hot, bright conditions, and converting its photosynthetic machinery to the more efficient C4 pathway used by maize or sugarcane.
Potentially boosting yields by, what, 50%?
That's the theoretical target. It's a massive undertaking, requiring changes to dozens of genes, coordinating expression across different cell types. But it's an active area of research, only possible because we can integrate all this omics data.
What else?
De novo domestication. Going back to wild relatives of crops, which often have great stress tolerance but terrible yield or plant shape, and using our knowledge to rapidly redomesticate them, but keeping the useful wild traits. Maybe changing plant architecture for denser planting.
Replaying domestication, but faster and smarter.
Kinda, yeah. And then there's synthetic biology, combining pathways or traits from different species in novel ways.
The pomato idea. Potato tubers and tomato fruit on one plant.
That's the futuristic poster child. Maybe not practical yet, but it represents the ambition-building entirely new biological systems based on our detailed understanding from genomics and omics.
It's an incredible technological explosion. Sequencing whole pangenomes, measuring thousands of metabolites, it feels almost overwhelming. But at the end of the day, what's the biggest bottleneck? What's the final frontier here?
It's not generating the data anymore.
Mm.
We are drowning in data, frankly.
Yeah.
The real challenge, the ultimate success of all this for future breeding, hinges entirely on our ability to integrate it effectively.
Bringing it all together.
Yes. Standardizing it, building models, creating predictive algorithms that can take all this diverse information, genetic variation, expression levels, metabolite profiles, and accurately forecast how our plant will actually perform in a complex variable field environment. That integration, building truly predictive models of biological behavior, that's the grand challenge, and honestly the most exciting frontier ahead.